Thank you again. I'm Pedro. I'm again a PhD student at Gagneur Lab, and today I'm going to present you how we can leverage DNA language models to look at nucleotide dependencies in the genome and reveal functional elements. So language models in DNA are usually trained by predicting or reconstructing a nucleotide from a specific sequence context by assigning a probability to each one of A, C, G, or T for each position. And we and others have shown in the past that the reconstruction from the language models highlight functional elements in the genome. So this is an example of a stretch of DNA in yeast, which contains a tRNA and some flanking regions. And this is the reconstruction for each nucleotide and position by our model species LM fungi. As you can see, the reconstruction is highest inside this regulatory element. And that's presumably because the identity of each nucleotide is more constrained here than in the flanking regions. But for the models to be able to predict a nucleotide inside the tRNA, they have to understand the dependency between that nucleotide and the specific context they are in. So we go one step further and we check not only the reconstruction of each nucleotide, but how a reconstruction of a target nucleotide changes given substitutions on core nucleotides. So in practice, if we look again at the example of the tRNA, and we select one query and one target nucleotide, and that in this case are quite distal, and you might know that the tRNA falls in a specific structure, and here is the secondary structure, and in this specific case, the query and the target nucleotides are in Watson-Crick base pairing. So we have U and an A, or as you might know, the U in the case of RNA and belongs to a T in DNA. And the exercise we do is the following. Like if we were to mutate the query nucleotide, which is a T, to each one of the possible alternative 3 nucleotides, what would the predictions of the model look like for the target position for each one of A, C, G, and T? So for example, if we mutate a query nucleotide to an A, then the model is going to favor the prediction of a T. And it's quite interesting because we know this region has to base pair, so the model is reflecting this base pairing constraint. If you look, for example, at the C, then the model will favor at the target position a G. And if you look at the G, the model will favor the C and the T. And as you might know that in RNA, there is also this wobble base pairing between the G and the U. So this is also highlighted here in this table. And then we extract a value per query and target nucleotide, and we call it dependency. And this value is basically the maximum log odds ratio in this table. So for each one of the possible query and target nucleotides, if we get a value, so if you have n nucleotides, you'll get n squared dependencies. And then we plot it in the shape of a map where each query nucleotide is a row and each target nucleotide is a column. So if we go back to the previous example, we see the T in the row here and the A in the column here, and the dependency will be the maximum value in this table. So from just broadly looking at the map, we see striking patterns. So we can see these anti-parallel diagonal patterns, and they actually map to each one of the arms of the tRNA. So these stretches of contiguous base pairing in the tRNA. And the tRNA does not fold only in a specific secondary structure, it folds in a tertiary structure also. And we see, for example, this dependency, which is highlighting contact, which contributes to the classic tertiary structure of the tRNA. So to conclude, just from these simple observations or querying of the model, we are able to obtain without any fine-tuning these functional interactions without having a model that particularly focuses only on tRNA loci, without any kind of sequence alignment and without any biophysical rules. So this prompted us to broadly analyze dependency patterns across different genomes across different DNA and RNA language models, both developed in our lab and outside. And broadly, we can categorize the patterns into two main groups. One is this kind of local dependencies, like on-diagonal patterns and block-like patterns. And these distal dependencies, like anti-parallel dependencies, like we saw in the structure, some parallel dependencies, and other kinds of off-diagonal blocks and asymmetries. So starting with these local patterns, let's look at a promoter region in yeast. Here is the reconstruction by the species LM fungi language model. And as you see, the reconstruction is already highlighting different transcription factor binding motifs to the level of their PWMs, and also highlights this poly-DA region. And if you look at the dependency map, you see that these transcription factor binding motifs appear as these very dense block-like regions. And that's because in a motif, all bases are bound by a single factor. So substituting one base will effectively change the meaning of the whole motif, and that's captured by the model and the dependencies. And that is in contrast with other highly reconstructed regions, like this poly-DA region, which and because of this, we actually thought perhaps we could develop. We developed with this score, which is the block score, which measures how block-like each position along the diagonal is, how dense the dependencies are. And we use it to classify whether a nucleotide is bound by a transcription factor or not on a data set with the ChIP-seq from multiple transcription factors in yeast. And here is the performance. So on the blue line is the block score. And these we can see that the performance is better than taking the reconstruction at the nucleotide or taking the conservation here obtained by the fast-cons score. And is actually at the level of the performance of the PWM, which has been experimentally determined. And another thing we should add is that the positive set is actually slightly biased towards the PWM because the positive set was defined by ChIP-seq peaks, which simultaneously have a PWM match. It's quite impressive that a purely unsupervised score can already be comparable to this PWM score. But we don't see only dependencies or blocks within the main diagonal. We also see blocks off diagonal, which reflect interactions between motifs. So here is the example of splicing and the intron and the flanking exons. And in splicing, in the intron, we have these specific characteristic motifs, the donor, the acceptor, and the branch point. And you can see a dependency between the donor and the acceptor and the donor and the branch point. And in introns across yeast, dependencies between the donor and the acceptor are higher than dependencies between donor and decoy acceptor, so sequences that look like an acceptor but are not a canonical one, or dependencies at matched distances. And this is not only true in yeast, but also true in human. Dependencies are also seen between donors and acceptor. We took this model called SpliceBird, which has been trained in different vertebrates, including human. And here is an example of a splice region where we can see from the RNA-seq track, there are two exons, and there is one individual which has a variant in a donor which induces exon elongation. And this can be actually spotted by just the dependency map. So you see an interaction or a dependency between the donor and the alternative donor, indicating that substituting the donor will change the predicted probability for the alternative donor. And in fact, dependencies between a variant and the corresponding outlier junctions are higher than dependencies at matched distances for different categories of where the variant can be. So regardless of whether the variant is inside the intron, in the splicing regions, donor acceptor, etc. All right, so other kinds of distal dependencies we see are these anti-parallel dependencies, like we saw in the case of the tRNA. And in order to evaluate them, we have just developed very simple convolutional filters to scan these dependencies across the genome. And once we scan the genome, and specifically mostly using this model called ReNalmo, which has been trained on millions of non-coding RNAs, many coming from our farm, which contains a lot of structured RNAs. We find that the dependency maps are already highlighting basically essentially the contact maps of these structures, and even very complex structures like this one, which include not only like the classical stem loop contacts, but also non-nested contacts such as pseudoknots, which are known to be hard to model computationally. And in fact, if you take just the dependencies to score secondary structure contacts, you can already do a pretty good job across different families of RNAs. But another thing that we have found is that there are several false positives in the dependency maps that do not belong to the secondary structure, but actually belong to non-canonical structural contacts. So this is an example in an RNA. Here you have the ground truth annotation. And in black, you see the canonical secondary structure contacts, and in gray, these non-canonical contacts that contribute, for example, to the tertiary structure of this RNA. And you see they are already reflected in several dependencies. But if you look at this fine-tuned ReNalmo model, so the model we used to compute the dependencies was the pre-trained DNA language model. But the authors of this model have also further fine-tuned their model for secondary structure prediction. And if you employ this model on this RNA, you see that essentially the non-canonical contacts are not predicted. And this is also seen across multiple non-canonical contacts on a Comparna PDB database. So this highlights the fact that the dependencies can highlight some functional contacts that have been learned by the pre-trained model, but are eventually lost by the fine-tuned of the model. And all of these results were on known structures, and they were already quite impressive. So we thought that perhaps we could find some novel structures that were not being able to be found by classical methods. So we scanned the genome of E. coli for anti-parallel dependencies that were in regions that were not annotated as being a structure. And we partnered with Dani Incarnato from the University of Groningen, who performed this assay, which is the DMS map-seq, which measures the reactivity of each nucleotide to DMS, specifically A and C nucleotides. And the higher the reactivity of a nucleotide, the more likely it is in an unpaired state. And we found 4 novel structures in E. coli, and this is one of the structures we found. And as you can see, the several anti-parallel dependencies map to different stems. And we also see, for example, this anti-parallel dependency, which belongs to this stretch of A's interacting with this stretch of U's, and is supported by the DMS to be a pseudoknot. All right, but we don't get only anti-parallel dependencies, but we also see these parallel dependencies, and especially we see them in repeats. So here is a region which contains a data box and a sequence element and its repeat right afterwards. And you see, contrary to this data box, which appears like block-like, similarly to what we have seen before, here within the repeat, we have this parallel dependency, and that stems from the fact that substituting the first element of the repeat will have an influence on the first element of the second repeat, and so on and so forth. So we have this one-to-one correspondence. This is quite intriguing because it led us to believe that perhaps the model is making its prediction not based on a memorization of this element, but based on the fact that it is copying. So perhaps the model has learned a copying operation. And to explore this, we have just simply developed random repeats that the model has not seen on the training set, and have included them at different distances. And indeed, the model reconstructs them quite well and also highlights them as parallel dependencies. And the longer the repeat, the stronger the dependencies. And this also works for inverted duplications, so repeats, which are the reverse complement of one another. So we have seen how these dependencies were able to give us insights into what the model has learned, but can we actually use them to also compare between models without any kind of fine-tuning? So for this, we selected this data set, which is the prediction of tRNA structure in human. Because most models have been trained, including human sequences, and many models have been trained only in human. And tRNAs, because tRNAs are some of the most conserved RNAs. They appear hundreds of times. They are conserved across species, and their interactions are also conserved. So if we take, for example, Rinalmo has been trained on millions of non-coding RNAs. Many come from structural databases. We see that dependencies already capture quite well the secondary structure. The species LM fungi, which has been trained only in fungi, so has never seen a human sequence, also already performs quite well. And then we see models like EVO, has not been trained in eukaryotes, that can highlight some anti-parallel dependencies, but we also see some artifacts. And this stems from the fact that EVO is an autoregressive model. So EVO predicts the next token based on a previous context. And that induces an artifact where kind of EVO only realizes it is inside the tRNA once it has seen enough nucleotides, and only then it can feel the dependencies. And this can be mitigated by running the model on the forward and the reverse strand, as you see here. But you can still see this artifact. For the nucleotide transformer, it has been trained with input non-overlapping 6-mers. And that seems to introduce a special artifact where the nucleotides inside these 6-mers artificially depend on each other. So this is true locally, but this doesn't seem to be true distally, as you see some more fine-grained dependencies in these antidiagonals. And then models like Hyena don't really show any kind of dependencies in the tRNA. And actually, the big divide here in the models is not exactly their architecture, but it's rather their training data. So especially models that have only been trained in human perform much worse at this task than models that have been trained in multiple species, regardless of whether these species include human or not. And to finish, I'd like to show how we can use dependencies to score variants in the genome. So there are several studies which leverage DNA language models to score variants by essentially comparing the reconstruction of the variant with the reconstruction of the reference nucleotide with a log ratio. But here we developed the variant influence score, which quantifies how much a variant affects the probability of all the other nucleotides in aggregate. And we first focus on a data set, CleanVar pathogenic, which contains pathogenic and benign variants, and we subset it for non-coding variants. And as you can see, the influence score can already broadly differentiate between benign and pathogenic variants. However, well, if you look at its performance, is better than taking the log ratio, which is based on the reconstruction of the variant versus the reconstruction of the reference, but it's not better than taking other kind of traditionally used scores that are based on alignment and conservation, like PhyloP and FastCons. However, one has to say that the way this CleanVar defines pathogenic and benign variants is also helped by these scores. So there is a slight bias here. So we decided to focus on a different data set. And this data set is mutagenesis screen in 9 human promoters from Kircher et al. And here you see an example of a chunk of the human LDL receptor promoter. and the nucleotide reconstruction probabilities by our model, species LM metazoa, and the results from the mutagenesis screen. The specific example, they seem to already agree quite nicely. However, if you check the correlation between, for example, taking the reconstruction or the log ratio and the variant effects, it's still not better than taking the scores from PhyloP and FastCons, which are based on alignment and conservation. However, if you take the influence score, so the effect, the influence of a variant on the reconstruction of all the other target nucleotides, you can do much better at this task, way better than taking the log ratio from the same model, and better than the other alignment-based conservation methods. All right, so also to put this into perspective, we have also wondered what, for example, the performance of state-of-the-art supervised models would be at this task, although it is not the purpose of this project really to compete against supervised models. We wondered what would the performance be here. And if you run Borzoi, the state-of-the-art model to predict that predicts RNA-seq profiles and in extension RNA abundance, the performance of Borzoi is slightly better than the influence score, and that is to be expected because the influence score, well, it captures all kinds of interactions, be them, for example, splicing structure or expression, and it's not based on any experimental data. And Borzoi has been trained to predict, for example, RNA abundance, and can be adjusted to the specific tissue where these promoters have been tested. So it's quite impressive that the performance is not so far off. And perhaps more interestingly is that combining these scores with a simple linear regression that weighs them per promoter can achieve a better result at this task. So this hints to the fact, at least from this benchmark, that it is possible that DNA language models can complement state-of-the-art supervised model predictions. So to wrap up, our observations have implications on variant interpretation. So nucleotides' influence on reconstructing others rather than its own reconstructability is a better predictor of function. In regulatory genomics, so we have seen that, for example, transcription factor binding motifs appear as blocks, but for regions in the genome that are less well studied and non-model species, so we can perhaps help with this kind of visualization and also for RNA structural biology. So we have seen that the dependencies recover functional RNA secondary structures and beyond, so pseudoknots, alternative structures, tertiary structure. And for computational biology, so we have essentially established a way to compare between language models without fine-tuning. And last, we also have provided a new way to visualize sequences. Actually, several insights and analyses were inspired by just looking at the different patterns we see in different maps. All right, I would like to thank the whole team that has been working on the project, especially to Alexandra Cajolas, which is co-firsting this project, to Dani Incarnato for all the experimental work and help on the structural analysis, and to Julien Gagneur for all the supervision. And I'll finish up with this very cool map in a particularly busy region in the mitochondria. All right, thank you very much, and I'm open to any questions.